Welcome you all. In today's lecture, we will discuss storage lipids. Triglycerides or neutral fats represent the predominant form of storage lipids found in the organism. In plants, there is a preference for storing neutral fats within seeds due to their higher content of reduced carbon, offering a more energy dense source compared to carbohydrate. Unlike carbohydrates, which retain water, neutral lipids are anhydrous and thus stored without water. This characteristic helps in reducing the weight associated with water retention, making seeds lighter and more suitable for dispersal. Triacylglycerols, the primary type of neutral fats, consist of glycerol esterified with fatty acids at all three hydroxyl groups of glycerol. This arrangement enables efficient storage of energy in a compact and weight efficient manner within seeds. Glycerol with its three hydroxyl groups can undergo esterification with either similar or different fatty acids. When all three hydroxyl groups are esterified by identical fatty acids, the resulting compounds are named based on those specific fatty acids such as tripalmitin, tristyrene and or triolene corresponding to palmitic acid, stearic acid or oleic acid respectively. However, if glycerol's hydroxyl groups are esterified by two or three different fatty acids, the nomenclature specifies the name and position of each fatty acid. This specification is achieved through stereospecific numbering. It is, in short, it is SN, where SN1 refers to the top carbon, SN2 to the middle carbon, and SN3 to the bottom carbon atom of glycerol. Neutral fat synthesis in plants primarily occurs within the endoplasmic reticulum membranes. Once synthesized, these neutral fats are typically stored in the oil bodies found within the endosperm cells of seeds. However, there are exceptions where triglycerides may be stored in other plant parts such as fruits, stems or roots. For instance, in Tetrina mangolica maxim, commonly known as oil firewood, stem tissues serve as storage sites for triacylglycerols comprising approximately 9% of the plant's dry weight. Another example is Manihot esculenta, where triacylglycerol accumulation predominantly occurs in roots, constituting around 25% of the dry weight as oil. Additionally, in many heart esculenta tubers, both oil and starch are accumulated, with oil accounting for approximately 30% of the dry weight. These instances illustrate the diverse storage patterns of triglycerides in different plant species and organs, showcasing the versatility of plants in adopting their lipid storage strategies based on their specific physiological and ecological requirements. This slide is about waxes. Waxes also serve as energy stores. It represents intricate molecules composed of a blend of long chain hydrocarbons, alcohols, aldehydes, acids, and esters of long chain alcohols. Biological waxes form through the esterification process, combining saturated and unsaturated fatty acids ranging from C14 to C36 with long chain alcohols spanning from C16 to C30. Typically, the melting points of biological waxes fall within the range of 60 to 100 degrees centigrade, often exceeding the melting points of triacylglycerols. In the biological realm, waxes play a crucial role as the primary storage form of metabolic fuel in plankton, which are small free-floating microorganisms. Characterized by their highly hydrophobic nature, waxes possess remarkable water-repellent properties. Vertebrates utilize specific skin glands to secrete waxes, providing protection to hair and skin while ensuring flexibility, lubrication, and water resistance. Birds, particularly waterfowl, produce waxes from their preen glands to enhance the water resistance of their feathers, showcasing the multifaceted roles of waxes across various organisms in nature. In this slide, we will discuss about that how the leaves are coated. Shiny leaves coated with substantial layers of waxes are a characteristic feature of several plant species, including holly, rhododendrons, poison ivy, and numerous tropical plants. This protective coating serves multiple functions within the plant's ecosystem. Firstly, it acts as a barrier, inhibiting excessive water evaporation from the leaf surface. This is particularly crucial in environments with high temperatures or low humidity, where water loss through transpiration can be significant. Secondly, the wax layer provides defense against parasites and pathogens, creating a physical barrier that makes it difficult for pests to penetrate the leaf surface and cause damage or infection. Waxes are intricately integrated into the cutin network of the plant's epidermis, forming an amorphous substance that coats the outer layer of the leaf. This integration enhances the impermeability of the cuticle to water, thereby reducing water loss through evaporation and helping the plant maintain its hydration levels. Additionally, the presence of waxes on the leaf surface can make it more challenging for parasites to establish a foothold, further enhancing the plant's resistance to biotic stresses. Beyond their roles in plant physiology, biological waxes have diverse application in various industries. In pharmaceuticals and cosmetics, for example, waxes such as lanolin, beeswax, carnauba wax, and spermaceti wax are prized for their unique property. Lanolin, derived from lamb's wool, is commonly used in skincare products for its moisturizing properties. Beeswax serves as a natural emulsifier and thickener in cosmetics and is also utilized in candle making. Carnauba wax, extracted from the leaves of Brazilian palm tree, is valued for its glossy finish and is commonly found in polishes, waxes, and coatings. Spermaceti wax, obtained from the head cavity of sperm whale, was historically used in candles and ointments, although its use has diminished due to conservation concerns. These biological waxes exemplify the intersection of natural resources with human ingenuity, offering a multitude of practical application across different sectors. Waxes are synthesized within the membranes of the endoplasmic reticulum or ER by enzymes called wax synthases and are subsequently transported to the cell wall. The process of exporting waxes across the plasma membrane involves specialized proteins known as ABC transporters. Although the exact mechanism by which wax monomers are transferred from epidermal cells to the plant surface is not fully understood. It is believed to involve lipid transfer proteins. In plants' aerial organs, a significant portion of fatty acid is allocated towards the synthesis of both wax and cutin, contributing to the protective coating on the plant surface. Some plant waxes hold commercial importance, such as carnauba wax found on the surface of the Brazilian palm tree. Another noted example is the jojoba desert plant called the Simonsia chinensis, which stands out as the only angiosperm utilizing wax for storage purposes. The liquid wax extracted from its fruits is highly valued in the cosmetic industry for its moisturizing and emollient properties, highlighting the diverse applications of plant derived waxes beyond their protective roles in nature. As you can see, this figure 1 shows the primary constituent of bee wax, tricontinyl palmitate, which is formed through the esterification of palmitic acid with the alcohol tricontinyl. Bee wax exhibit notable characteristics such as its solidity at room temperature and is complete impermeable to water. Moreover, the figure B shows a honeycomb which is constructed from bee wax maintain its firmness at 25 degrees centigrade and effectively repels water. The term wax has its origin in Old English, specifically derived from wax, which refers to the substance composing the honeycomb. This linguistic connection underscores the historical association between wax and its natural source, highlighting the cultural significance of bee wax.